Hi, everyone. Thanks for coming to today's iGEM Toronto Dry Lab overview. So welcome, everyone, especially those who aren't part of iGEM Toronto. We're trying something new this year where we're inviting everyone who are interested in what we're doing to get more involved and be more aware of the type of work that we're doing. And if you're interested, definitely feel free to reach out to me or Joseph, who's also on this call. You can reach out to iGEM's email as well to follow up. And if you're interested in contributing to some of our work, we'll be more than happy to chat with you. So last week, we gave an overview on iGEM Toronto Wet Lab, where Joseph gave a very nice and detailed explanation on our project, an introduction to our project, and what the wet lab does with respect to this synthetic biology project that we're working on, which is enzyme design. So if you haven't seen that, it's on our YouTube channel. Just search up iGEM Toronto. It's the latest video there. Today, I'll be mostly talking about the dry lab side. And now this is joint work done by, let's see, me, Alston, Isaiah, who are the core dry lab members. And we also have Helena, who's been coming to our meetings, and Yinshi. So let's get started. What do we do as part of the dry lab of iGEM Toronto? We generate enzymes using machine learning. So in this picture, the orange mesh is an enzyme, and it's a space volumetric filling version of this enzyme. What we want to do actually with our enzymes, the particular enzyme we were working with is a plastic-degrading enzyme. So this grayish black blob in the middle, that's a plastic molecule that is very nicely docked into the structure here. So this is essentially physically how this process works. You have an enzyme, you have these polymers of plastic that interact with it, and if parts of it, it's going to cut here and break polymer down into its this is one particular example. How do we do this enzyme design? Evolutionary scale modeling. This is a bit of a complicated slide, but essentially what we're doing is we use this model called ESM for short, so ESM, and it gives you a probability of any protein sequence. And the probability here I show as log likelihood, which is to say the higher it is, the more likely your protein is going to exist in nature. So here are a bunch of different generative models that we tried. Generative models are machine learning models that can generate new protein sequences. So for example, you can train for a GPT-2 model, if some of you have heard about that. It's usually used for language and natural language processing applications. But you can just think of a protein sequence as really a sequence of letters, sequence of amino acids, a sequence of words. So we can train each generative model on some distribution of sequences, and then we can score them. And this is exactly what we're doing. So in blue is our training distribution. I'll talk about that a bit later. But this is a natural distribution of their likelihoods. We have LCC, which is the enzyme that can degrade plastic. That has some number, right? That has some likelihood under this model. And we can evaluate how well our generative models do by computing the likelihoods of their generated sequences. So you can see some of these models are not so great. They have lower likelihoods than others. And some other models like GPT-2, CCMGen, they have a significant region that's at or above the likelihood for the enzyme that we care about. Of course, this is just a proxy. This is just one of the many things we're looking into. These models can actually filter millions and millions of sequence designs. So suppose you have some proposed sequence designs. Yes, Maya, this is being recorded, I believe. If you can confirm Well, is this being recorded? Can someone confirm that?
This is being recorded.
Great. So to filter millions of designs of our generated sequences, we can pass these sequences through this generative model, this ESM. For every sequence is going to give a number, which is the likelihood of that sequence. And this is a very efficient way then to filter out potentially millions of designs. Now, we already have tens of thousands, hundreds of thousands of designs. And this is exactly what we're doing, is that we're using such a probabilistic model to capture properties of sequence space. So this is a very broad overview. You're probably kind of confused right now. So let's go into a bit of detail here. All right. How do we do this? How do we actually use machine learning and design new proteins? That's a big question that we're hopefully trying to answer today. And if you have any questions, feel free to leave them in the chat or just unmute yourself and ask away. So ultimately, what we wish to model is a protein function landscape. Or it could look like something like this, where in our case, we care about the speed of catalytic activity, right? So it's the speed at which the enzyme degrades its substrate, the plastic ligand. Now, this function landscape can correspond to any desirable property you wish to measure. It could be something like thermal stability, binding affinity, catalytic activity, as I just mentioned, or free energy of the reaction. It's really diverse. You can think of any property you want. In this case, we care about catalytic activity. So every point along this landscape is a protein sequence, and the height of that landscape corresponds to, essentially, in our case, the rate at which an enzyme degrades plastic. So naturally, you want to climb this landscape to find a nice point, sort of climb the hill and find sequences with good function. Unfortunately, in reality and in practice, we don't have an accurate model of this landscape. In reality, we maybe have a couple of points, a finite number of points that sample this space very sparsely. And the objective for a machine learning model is to use this data that we somehow have from experiments or from experiments that we do ourselves, and use that to infer function for unseen sequences. Because once you have that, you can imagine scoring millions of designs, you either through rational design or computer generated designs, pass them through the score function, and reliably the highest scoring ones are going to be good, and you can just synthesize them in the lab and test them out. That's the pretty picture, but in reality, that's not the case. So the traditional workflow is you start from a protein sequence. Traditionally, you would align the sequence with similar sequences through something like a homology search, basically a similarity search through the database of all known sequences. You compare against them, find some similar ones that you already know the function of. Perhaps you also know the structure of, right? So you then somehow determine the structure from a sequence. Of course, in 2021, you can have AlphaFold 2 do that for you and give you an idea of what the structure may be. And under the traditional biochemist view, structure implies function. So sequence implies structure, which implies function. Now, again, function is any diverse property that you wish to measure. But for our machine learning models, what we actually do in practice is to skip the structure step. And you can go actually directly from sequence to function. In practice, how well does this work? Sometimes it works surprisingly well, but sometimes it's not so good. So this is still an area we're sorting out for this particular enzyme we're working with, and we'll have some more results to share in the future. Now, suppose you have this really good scoring function. You have a very accurate function that takes in sequences and gives you its catalytic activity. Unfortunately, that's not the end of the story, because you may have heard of this before, this incredible vastness of protein sequence space. So it's called combinatorial space, where at every position you have 20 possible choices of amino acids. And you can have all these 20 choices at any position along a sequence. Then your number of possible protein sequences completely explodes into a ridiculous number that you can compare with other large numbers, and you get a sense of how large this is. So the number of atoms in the universe, if all of them were doing computation at the speed of the fastest computer in the world, and all of them have been doing it since the beginning of time, multiply all these three numbers together, you would only be able to explore a tiny sliver of the possible space of protein sequences. Clearly, we cannot exhaustively search through this space. We need smarter algorithms and models that will help us guide the search through the space and focus it on promising regions of sequence space. That's exactly what the generative models we wished to train, and that's exactly what their goal is. So here's the same diagram that you've seen on the previous slide, but with all the arrows going in the reverse direction. Previously, you were going from sequence to structure to function, but that's expensive because you have to search through vast spaces of sequences. What if you go backwards, right? How about starting from a function? Let's infer the sequences that can have those desired functions. So each of these arrows is then a generative model. For example, the first arrow, given a structure, predict the sequence that can fold into a desired structure. This arrow right here, a bit more elusive. Currently, not really sure what machine learning models can do it, but definitely the Rosetta community is really good at this. Given a function, predict the structure that can accommodate that function. For example, you can imagine designing a backbone that can have shape complementarity to a desired binding target, like DNA or antibodies, etc. This is the arrow that we care about, this arc on top. Given function, predict the sequence. So this is sort of this elusive thing. Can we somehow predict sequence from can we somehow generate a distribution of sequences just by giving somehow to a machine learning model a somehow a desired set of functions? How do we do that? It's very elusive at the moment. Evolution actually gives us training data. Of course, it's not for free because hundreds and thousands of scientists have spent decades curating sequence databases for us. We're just leveraging all the hard work that they have been depositing into NCBI. And essentially what we do is a blast search with a wild type version of the enzyme we wish to design. And it's going to search for similar sequences. So on the right, you see a sequence logo. This is something that members of the dry lab stare at every single day. And this is what we dream of when we go to sleep. You can see that every position has a combination of letters. The higher a letter is, for example, at position 207, it indicates that position is highly conserved for one particular residue. So here, histidine is pretty much the only residue at position 207. Similarly for some of these other conserved positions. Some other positions where it's not so high, that's more variable. Now, what is the takeaway from this picture? The takeaway really is that the fact that evolution, which is this random process over hundreds of millions of years, can somehow preserve certain positions but not others, implies that these conserved positions are important for protein function. Now, it's slightly unclear what aspect of function they're important for, but we just know it's important, right? These are the statistics of sequence space that we wish to model. Now, the hypothesis then is that similar sequences have similar structures and similar functions. So you have a homologous set of sequences. Ideally, this is what SciBlast will return to you, is a homologous set which supposedly can derive from one ancestral sequence. Then through process of evolution, parts of the sequence have diverged, but other parts of the sequence are still conserved. And those conserved parts of the sequence will give the protein its desired function. All right. So this is our training data. To drive home the point of the power of such search algorithms, I'm actually doing here a structure alignment of two plastic degrading enzyme sequences. So LCC is an enzyme that we're working with. PETase is another enzyme. In fact, these two are homologous. They're only actually about 48% identical. So about half of its residues are different from each other. But if you look at the structure of these two enzymes, they're practically the same. And you can see I've highlighted here in red and magenta the active site of both proteins. The active site gives both proteins and both enzymes its ability to degrade plastic. And you can see how strikingly these residues are aligned in terms of their geometry. So hopefully, this BLAST search gives us a distribution over sequence space such that when we train a generative machine learning model over it, we essentially will model this distribution right here, P of sequence given function, because we're assuming that similar sequences have similar function, and thus this is the distribution we're modeling. Okay. Now, to do that, our machine learning models need to take sequences as input. So I'm here just showing you the first 3 entries of the BLAST search that we did. And you can see quite, strikingly, some of these sequences are quite similar. So BLAST is this really powerful tool for sequence similarity search. And you can see like this is a completely different organism, right? But the sequences are quite conserved. So we have 5,000 sequences from this search. We train our machine learning model on it. How do we do that? The sequences are encoded through either one-hot encoding or tokenization. They're pretty similar, right? These two things, one-hot and tokenization, is also a very simple idea. You instead of having a string of letters, you convert that to a string of numbers that the computer can take in and ingest into a model. So here I'm just listing these amino acids as Python list. And what you can just do is convert each letter into an integer. That's exactly what these two dictionaries do down here. So for example, this sequence with 6 amino acids is represented by this matrix with 6 rows. Now each row has a 1 at the index corresponding to that amino acid. So for example, this matrix here is MNFPRA. You can match where the 1s are with the position of that letter in this list. So this is the input to a machine learning model. And in fact, you can train entire sequence to function models just based on this representation. And sometimes you'll be surprised how well this works, even though it's a super simple representation. All right. How do we train models? This is an interesting point. What we want is generative machine learning models that can recover and model a desired distribution over sequences. We train these types of models in an unsupervised way. Unsupervised just means providing no labels to the model. All right. So there are 3 ways of training these models that I'm showing here. The first way is autoregressive, where given all the amino acids that came before it, predict the next one. So here in this example, you're given M and N, and you predict what goes in the green one, right? What does it mean for a model to do a prediction? Basically, it just outputs a bunch of numbers, right? 20 different numbers. Each number is interpreted as the probability of some residue being there. So in this case, for example, you have serine and threonine being quite probable at this position. So the model's output is a probability distribution over possible amino acids. And then to generate new sequences, you basically sample from the model output distribution and sample 1 letter at a time. Right? You sample this one and say, oh, this is a serine, great. And then you go on to the next step. What is the next letter? And so on. And doing so, you can actually generate entire sequences from left to right. But then you may be asking, proteins are not like sentences. They don't necessarily have just a left to right direction. All right. So another training approach is bidirectional, where you're not only given amino acids to its left, but also to its right. Yes, but also to its right. So this sort of is a slightly more appropriate for proteins because the identity of amino acid may not just completely depend on everything to its left. It may also definitely depend on things to its right, right? But that's also sort of a sentence, a natural language way of thinking about sequences. A really interesting way of thinking about protein sequences and how to train these models to generate new sequences is this third way, the structure-based training task, where given sequences, given the residues that are structure neighbors of the missing residue to predict, given those as context, predict the missing residue. Now, this is very biologically motivated because you can imagine the identity of amino acid probably depends on its local amino acid environment, particularly for very buried residues where there's not a lot of space for the side chain to move around. That place, the identity of that residue is just definitely influenced by its surrounding neighbors. All right, it may not be really influenced by like the first couple of residues there. Essentially, all three training approaches, what they do is that you mask out particular tokens, or in this case, residues along the sequence, and you ask the model to recover what was supposed to be there in the wild type. So why does this work at all? Well, it works because the distribution you're training on is a set of known sequences that are known to function in the wild. And this means that it's trying to recover a distribution of sequences that are known to work. All right. So ideally, if your model is actually doing this training correctly, sampling from such generative models should give you sequences that fold properly, which is the first criterion, and also have similar function, which is the plastic-degrading property that we care about. Great. Instead of training these models from scratch, we can actually leverage these pre-trained models that people have already trained on 25 to actually over billions of protein sequences. So 25 million to billions of protein sequences, essentially all of known sequence space. And one such example is this ESM model, this evolutionary scale model. And I want to show you some interesting results here, right? That doesn't necessarily, isn't necessarily intuitive, but it's sort of what happens when you work with something that no one has done before, is you don't really know how to make sense of this data. So ESM, for example, is a probabilistic model. It assigns a score, P star of X, for any sequence X. I put a star there because it's not a real probability. It's not a real likelihood. It's actually a pseudo likelihood, right? If you're interested in why it's called pseudo, yeah, that's for another story. So it assigns some score, right? You can rank sequences relative to each other. That's great. And these scores capture the likelihood of a protein sequence under its distribution, which is all of protein sequence space. And some people have found in the literature that these scores can correlate with protein fitness. Now, this is now very interesting to think about. If you look at the two plots on this slide, one is I'm plotting the pseudo likelihood of sequences against thermal stability. The other one I'm plotting against catalytic activity. So the interesting thing with this ESM pseudo likelihood, for example, thermal stability. In fact, this is actually a negative correlation. So the more thermal stable you are, the lower your likelihood score. And now that might be weird to you because you think, oh, more stable should mean more likely under your model. My hypothesis to why this trend is occurring, a Spearman correlation of like 0.63 actually, so it's not a very weak correlation, right? It's there, the correlation's there. I think a nice hypothesis is to really think about what these generative models are doing. These models are trained on protein sequence space that exist in nature. But if you look at these high values of thermal stability, this is basically saying it can work. Like half the protein can still work at about 95 degrees Celsius. So you can think about this as sort of like, well, maybe nature hasn't really necessitated the need to evolve enzymes that function at such high temperatures, or perhaps there exist enzymes, that or plastic-degrading enzymes that exist at such high temperatures already that we haven't discovered yet. That's not part of our training set for a generative model. So this is also sort of counterintuitive. Then you would imagine the higher the likelihood, the better your sequence. But actually, in this case, it looks like the lower the likelihood, clearly the more novel your sequence, right? But it could also mean the more thermal stable. And so one of the conclusions here is that the likelihood score may be sufficient for protein function, but it may not be necessary. And so that might be something really interesting to explore in our wet lab studies. Something else that we can consider is protein structure. We have the structure, the 3D structure of the enzyme we're designing. And this is the structure right here. Why do we just throw this information out, right? Why do we draw a big X over it when we have access to this? So this year, we're not going to throw it out. We're going to leverage this 3D information. And what we're going to do is to train a machine learning model that basically does this stuff right here. Given the local amino acid environment, predict what goes into this missing spot. And you do this for all of the residues along the sequence. And then this is what you get. So such a machine learning model, what it does is to generate sequences that fold into a desired structure. And this is this particular distribution that we're modeling. Given structure, predict sequence, right? This is a distribution of sequences that can accommodate, that can fold into some particular structure. The model, one of the models we're using this year is John Ingram and others, they call it the structured transformer. So they adapted the super famous NLP model called the transformer for protein structures. And this is a logo of sequences generated if you give it the protein backbone of the LCC enzyme that we care about. And you can see, by the way, people of the dry lab, we've practically memorized where the conserved residues are at this point. You can see this histidine here is conserved, this aspartic acid here is conserved, and this serine here is conserved. That's the catalytic triad, I believe. But actually, this model has some other very wild sequences as well. For example, here, I think in the blast, it was a very conserved tryptophan here. But here, it doesn't show up at all. So we'll see what happens with these generated sequences. Obviously, the final confirmation is with the wet lab. That's great. That's great and all. We can generate sequences. All right. We can train generative machine learning models to generate hundreds of millions of sequences. Potentially, we can also score these sequences. All right. This is actually turning out to be a bit harder than we thought. So we're still working on this part right now. But how do we actually optimize protein function? Because that's what we really care about. If you do this generative machine learning model, if you train it on the blast sequences, all you're modeling is just this natural distribution. I mean, yes, it will be awesome if the generated sequences actually fold and function. But that's just a natural distribution. You're not going to get some revolutionary enzyme that's 10x or 100x faster just by training generative models and not doing anything with it. So this is the procedure that we think can optimize protein function, and this is formalized by David Brooks in one of his really, really amazing papers 2 years ago. So you generate sequences, you score them. That's fine. The third step is look at the scores of the generated sequences, take the best scoring ones, and then feed that back in to our training set. Then essentially we are shifting the generated distribution to regions of sequence space that are more promising, so that have higher function. And you'll notice this is a closed loop. So this is going to iterate for a couple of times. And every time it's going to be conditioned on optimal sequences. This is the algorithm called conditioning by adaptive sampling. I'll just go very high level over this. Definitely read the paper if you're interested in it. And hopefully it makes sense, although there's going to be a lot of symbols. I apologize in advance, but I think it's insightful and helpful to look at what we're dealing with here. We're going to encode a protein as a one-hot vector. This is just the space of natural numbers, and you have a protein sequence of length L. So you have L numbers, all right? We talked about this in the previous slide. X here represents a protein sequence. The goal is to model a distribution of sequences conditioned on our desired properties and prior over sequence space. So there are two things going on here. One is S and one is theta. S is the set of desired properties, and in our case, high catalytic activity. Theta is the space of known sequences. Right? It's some sort of parameterization of it. It's saying, well, let's look at all those sequences existing in nature, and let's try to model that distribution first. All right, let me expand it with Bayes' rule, and I'll hopefully try to make more sense now. So after you expand with Bayes' rule, you get this expression here, but really it's composed of two terms. One is this term right here, right? This P of X given theta. That's the distribution of sequences in your prior distribution. So this is your training data from evolution. You do a blast search. This is the space of known sequences that can function and fold in nature. And this is our initial sequence generator because this is P of X. This is a distribution over sequences from which you can draw samples from. You need an oracle, which is this term, P of S given X, where S is our set of desired functions and X is some sequence. So given a sequence, let's predict its function. Okay. Now this term is the thing that we want to model, but unfortunately it's intractable because of this denominator. Intuitively, what this denominator says is to take the sum, so this integral is to say, take the sum over all the X's. X's are protein sequences, right? This is pretty much impossible to do. You can't take the sum over all sequences. You can't do it with all the atoms in the world. Clearly, you can't do that on the computer. So to approximate this, we're going to use something, they call it the variational approach, which is this objective function here, where instead of modeling this thing directly with a neural network, we're actually going to use a neural network to model another distribution called a search distribution, which will closely approximate the target distribution we want to model. So this term right here, DKL, is this measure called the KL divergence, which measures similarity between two probability distributions. Now, what we want to do is to minimize this KL because it's saying you want these two things to be super similar to each other. The lower the divergence between these two distributions, the more similar they are, and that is our objective for our machine learning model. Ultimately, then we take the parameters that we get out of this minimization problem, phi, and these are the parameters of this Q, which is a machine learning generative model. After you do some math, this is the objective that you get at the end. So this objective is something that you actually want to maximize. Now, so before this was argmin, you want to minimize this, but after some derivation, you end up with this term, which you want to maximize. All right, unfortunately, this term is also problematic because of this thing right here, P of S given X. Why is it problematic? This is an expectation. If you expand this out, what this is saying is you sample some protein sequence from your initial generative model, and then you compute this term based on that sample. And you keep sampling 10,000, 100,000, a million times, and then you do that to approximate this expectation. Unfortunately, this term might be zero pretty much all the time. because this is saying what is the probability that your sampled sequence has the desired function that we're looking for. By the definition of design, none of your sequences in your training data will have that property you're looking for. Otherwise, you wouldn't need to do design in the first place. So unfortunately, by definition of design, this term would probably go to 0 most of the time. And that defeats our purpose of using machine learning at all, because it basically reduces to a problem of needing to sample 1 billion sequences and then computing this thing over all 1 billion sequences. How can we be a bit smart about that? We can use importance sampling, which instead of sampling over some prior space of sequences, we define another distribution R that if you draw samples from, these are going to be just the important samples that contribute to this term in here. Now let's try to parse this term a little bit, because this really motivates the next month or two of our dry lab research, because this is the objective that we want to maximize. Now, what are the terms in this objective? One is the prior. This thing is the sequence generator, right? This Q, the search model over sequence space that we're constantly iterating and improving. And then this is the oracle, right? A sequence to function predictor. What does it mean to maximize this term? It means that these things must be large, right? In particular, it's clear why this middle term has to be large. Given some sequence, you want it to be highly likely that sequence is going to have a desired function. That's clear. This thing is super interesting. You're saying for some sequence X, you want it to also have high likelihood under your prior distribution. Now we have full flexibility in defining what the prior is. And what is our prior in this case? Well, as I said, we can define our prior as the sequence space of similar sequences to our wild type with similar function. We can also define our prior to be the space of sequences with a similar fold, right? That's based on some machine learning model that takes in the structure and outputs this set of sequences. So here are our priors. And this is going to be part of our research for the next 1 to 2 months, is how to really incorporate some of these many generative models that we've tried and trained successfully over the last month and a half. How can we leverage that for design and optimization? Because why are these important? You can propose 100 mutations on your protein sequence, and your oracle, this term right here, might tell you this is like super good. But if you had like 100 different mutations, what if it disrupted your protein fold? What if it disrupted your protein function, right? And removed some of the really important residues for its enzyme function? That's what we really don't want. So to have these priors is sort of to have a reassurance that, well, our generated sequences not only have to be optimal under our sequence to function predictor, but they should also be likely to function under the set of sequences we know already function. And we should also know that it can fold into some desired backbone structure. So these are the 2 priors. We may add more priors as we go along, which we're thinking of, but this is a really strong starting point. Now, these priors, they're great, right? They're great in theory. They're saying, oh yeah, this machine learning model can score sequences that fold. But actually in practice, we have no idea if they actually do what they say they do, because very few of these generative models have actually been tested in the lab. So someone comes up with a brand new method saying, I can generate sequences that fold into a structure, but they don't have the money to actually do the X-ray crystallography experiments for all their generated sequences. So they use computational proxies, such as the sequence recovery rate and perplexities and so on. But really, we are sort of limited, and nicely limited, that if these models don't work, we've wasted hundreds of dollars sending sequences for synthesis. So we need positive controls. And we have some set of sequences that, like, for example, from here, generated from these ML models that have high likelihood under these initial priors that we should definitely send for synthesis and say, okay, if something has high likelihood under this generative distribution, is it actually a functional protein? And the experimental results will shed a lot of very insightful and necessary evidence into how much we can trust these priors, these generative machine learning models to filter our designs. So that's the first step. And now we're slowly moving on to the second step. So that's it for the presentation. And I welcome any questions that you have. I see some questions in the chat. Yeah, so thanks everyone. I will stop the recording because I think Miriam, you have an announcement that I'm sorry, I forgot that you want.